Hello everyone and welcome to Unacademy. So this is in continuation with the previous lectures which talked about the mechanism of urine formation. We have already discussed about the first step, which was glomerular filtration, in which from the blood the filtrate passes into the Bowman's capsule. In the next step we studied about the tubular reabsorption, that is from Bowman's capsule the filtrate passes into the PCT, and there are various useful products that are present in the filtrate which are reabsorbed back into the blood at various locations, be it PCT or the loop of Henle or through the DCT or even to the collecting tubules. So this is also important for our body because some useful substances are present in the filtrate that needs to be reabsorbed. Also, if our body is at a compromised state, that means when the water intake is less, then there are specialized hormones which can help us to recover, and the later parts, that is the DCT and the collecting tubules, are going to reabsorb water and release a highly concentrated urine. In normal cases we will be having a dilute urine, but when water uptake is less, a concentrated urine can be formed because the water channels inside the collecting tubule and the DCT opens up, which allows the water to move back from the cells of the DCT and collecting tubules into the peritubular capillaries. So now the third step and the last step of the mechanism of urine formation is tubular secretion. Basically, apart from various useful products, most of the wasteful products also enter the filtrate, but there are some wasteful products which remains in the blood. They have to be excreted out. So only 15 to 25% of the blood plasma is filtered out of the filtrate, but a lot of waste remains in the blood, in the peritubular capillaries around the renal tubules. So these wastes are actively secreted. Actively means they require energy against the concentration gradient, because anyhow they are present in low amounts in the blood, but their low amount is also to be secreted because they are of no use in our body. So actively they have to be secreted into the filtrate present in the PCT and the DCT. So this process is the tubular secretion process. This phenomenon of secretion of wastes is the tubular secretion process. So what all things are there? Excess of H plus and K plus. In this case, if something is in excess in the blood, then it would be passively transported into the filtrate. But as in case of creatinine, drugs, some pigments, and something like urea, all these might be present in small amounts, but they are not required by our body, so they can be actively transported out of the blood into the DCT or the PCT or any particular part of the tubule, the renal tubule. So now we will be just looking at what all things at various locations get reabsorbed and what all things get secreted outside the body. So this diagram explains it all in detail. First of all, in the PCT region, H plus ions and NH3. Before even starting with that, I would like to tell you that what all shown in pink is showing the secretion, tubular secretion, and what all shown in blue shows the tubular reabsorption. So now let's consider first of all the structure. We will revise the structure quickly. This is the Bowman's capsule, this is the glomerulus, this is the efferent arteriole which will form the peritubular capillaries over here, forming vasa recta over here, and peritubular capillaries again over here, and this is the afferent arteriole. Then we have the PCT, the thin descending limb, the thick descending limb, the thin descending limb, then the thin ascending limb and the thick ascending limb, and then finally DCT and the collecting tubules. So these are the various parts. Now H plus ions and ammonia ions are secreted into the tubule, but the hydrogen carbonate ions, sodium chloride and water are being reabsorbed back in the PCT region. So this is what happens in PCT region. Now when we come back to the descending limb, in this particular region water is being reabsorbed and sodium chloride is also being reabsorbed. So basically more amount of water gets reabsorbed here. Then Also in the thin ascending limb, sodium chloride is being reabsorbed, and secretion of urea is occurring. Urea is getting inside the tubules, and then NaCl is being reabsorbed back over here also, and over here also NaCl is getting reabsorbed. Now as I already told you, this is the inner medulla region. In this region, in the descending limb, large amount of water is being lost, thus this is the hypertonic location. But in this region, large amount of sodium chloride is being reabsorbed, it is the hypotonic location. So this is the difference between the two limbs. Then when we move further up in the DCT, NaCl and water are being reabsorbed, while K plus and H plus ions, which might be in excess in the blood, are being secreted. Finally, in the collecting tubule, urea can be reabsorbed. If a very high amount of urea is secreted, that is also not good. A minimum amount should be maintained in the blood. So some amount is reabsorbed. Then water can be reabsorbed in the presence of hormones, and NaCl is also reabsorbed. So this is all about the secretion and reabsorption. Basically, it depends upon the gradient that what all things are going to be reabsorbed. And also the hormones play a very vital role. Whenever there is a requirement of something in our body, then it is being reabsorbed. Similarly is the case with the salts. Sodium chloride is being reabsorbed whenever it is required, whenever the blood pressure has to be raised up. And if water is reabsorbed in case of some dehydrating conditions in the body, which happens only and only in the presence of certain hormones. Otherwise, the normal functioning involves removal of anything which is present in excess. But if you say the case of urea, it is being reabsorbed back, it is because a minimal amount of urea needs to be maintained in the blood. Blood cannot be without urea, it has to be there, some amount has to be there. So for that, a small amount of urea is reabsorbed back. So I hope you understood this process, and thanks for watching.